I think I'm going to start if it's okay for everybody. So thank you for joining this Flow Friday seminar series. Can you hear me still? Hello?
Yes.
Okay, so I can start. It's okay? Yes. Okay, so thank you for joining this flow cytometry seminar series, which is sponsored by the Cooperative Center of Excellence in Hematology. My name is Damien Reynaud. I am Associate Professor at Cincinnati Children's Hospital. And this presentation will focus on the hematopoietic stem cell, stem and progenitor compartment in mouse model and on the different way to analyze this compartment by flow analysis. So to give you a broad overview of this presentation, of this short presentation, I will first define this hematopoietic stem and progenitor compartment, particularly insisting on the functional heterogeneity which exists in this compartment. And I will also give an overview of the different nomenclature or flow cytometry signature which have been developed by many labs to take account of this functional heterogeneity and to put a phenotypic heterogeneity in parallel to this functional heterogeneity. I will also share the experience of my lab in analyzing this HSPC by flow cytometry. I will notably describe a 14-color panel that we developed to provide a comprehensive analysis of the early hematopoiesis. It's a ready-to-go panel that we use in our different study, and we can be used in different study if you analyze the impact of drugs, impact of a mutation, or the impact of different physiopathological condition on the hematopoietic compartment. So the principle of this 14-color panel is that we combine actually several nomenclature in one stain, several nomenclature coming from different labs in one stain, which is make it very easy to have a broad picture of the hematopoiesis in the mice, which is can save time and effort when you want to analyze a broad cohort of mice. We use also this panel to compare classical flow cytometry with spectral flow cytometry. Since 2020, my lab almost exclusively shifted toward the spectral flow cytometry aspect, which we found easier to manage. And finally, I will describe the future for this 14-color panel, as we believe that it can provide a base for future development if we want to integrate a new population, a new antibody, new nomenclature to grow again, to make this comprehensive analysis, if you want, more comprehensive. So to define the hematopoietic stem and progenitor compartment, as indicated by his name, it contains hematopoietic stem cells and hematopoietic progenitors. Hematopoietic stem cells are functionally defined upon transplantation in irradiated recipient. These HSC are capable of engraftment, and they are capable of long-term hematopoietic reconstitution. The classical definition is that these cells have to be able to ensure at least 4 months of reconstitution, which is a proof of their self-renewal ability. These cells have also to be multipotent, and they have to contribute to the reconstitution of all blood lineages in the irradiated recipient. The hematopoietic progenitor are downstream. They are also capable of proliferation and differentiation, both in vivo and in vitro, but they have limited capacity to reconstitute irradiated recipient. And when they do so, usually it's not for all lineages, and it's mainly very constituted the lymphoid lineage. Upon this very simple classification in 2 compartments, there is a great heterogeneity both in the hematopoietic stem cells and the hematopoietic progenitors that I try to show here. So the hematopoietic stem cells, people have show heterogeneity based on their level of quiescence in this population, their level of self-renewal, or their lineage potential. So, for example, people have defined dormant and active HSC. Active HSC are quiescent, but they are primed to cell cycle, and therefore are easily testable in a primary recipient, which they contribute to the reconstitution of the animal. Dormant stem cells, as indicated by their name, are far more quiescent, and usually they cannot be detected during the first transplantation, but their activity is then detectable upon secondary transplantation. People also have looked at the difference in self-renewal potential, and they find short-term, intermediate-term, and long-term HSC. Long-term HSC are the ultimate stem cells, which are capable of infinite reconstitution ability for many years. Intermediate-term HSC, it's a population or a compartment which have been defined by the laboratory of Norman Iskov, and they are thought to have a potential of about 6 to 8 months. Finally, you have a short-term HSC, which some of them have reached the 4 months threshold, so make them stem cells, but after this period, their potential fade and the reconstitution stop. The other characteristic that can define different stem cells, it's their lineage biased. So these cells are technically multipotent, but they can show some bias toward a specific lineage, myeloid or megakaryocytic. And also, or they can be fully balanced, capable of myeloid and lymphoid differentiation. And these have been defined also upon transplantation. So this is the heterogeneity in the stem cell compartment. You have the same thing, the same heterogeneity in hematopoietic progenitors. So the main difference in hematopoietic progenitor is that you can characterize the multipotent progenitors, or MPP, which are multipotent as the stem cells but have no or limited self-renewal capacity, at least below the 4-month threshold. Usually, upon reconstitution, this MPP give rise to mature cells for about 1 month, sometimes in some cases 2 months. So the multipotency has been also challenged in this as a little bit as it was the case for the stem cells. The multipotency of the MPP have been challenged, and now you have a definition of lineage biased MPP. So MPP which are multipotent but which are primed for specific lineage toward the lymphoid, myeloid, megakaryocytic, erythroid lineages. Below this multipotent progenitor, you have a set of lineage-restricted or lineage-committed progenitors, which have lost the ability to differentiate in a diverse pathway. So in this case, you have a lympho-myeloid prime progenitors, and you have all the classical restricted progenitor defined by the Weissman lab in the 90s, early 2000. So this includes the common myeloid progenitor, CMP, there is a mistake here, the common lymphoid progenitor, CLP, the common granulomacrophage progenitor, GMP, and the megakaryocytic erythroid progenitor, MEP. So this, you have a lot of heterogeneity, both in stem cells and progenitors, and it's functional heterogeneity, and these have been matched with a phenotypic heterogeneity, which can be detected by flow cytometry. So classically, all these HSPC are characterized by the absence of lineage marker, which are normally present in mature blood cells. As indicated here, when you analyze the bone marrow with a cocktail of antibody directed against this lineage marker, you have a large portion of the bone marrow which is positive, and a tiny portion of the population, 5 to 10%, which is lineage negative, and this is where the HSPC reside. The classical way to analyze these lineage negative cells is through the expression of two markers, c-KIT and SCA1. All the multipotent progenitor, which include hematopoietic stem cells and multiple MPPs, are present in the LSK compartment, which co-express KIT and SCA1, while the lineage committed HSPC are mostly present or exclusively present in this population, which express KIT but not SCA1, which is called LK here. So this is a very basic differentiation between this population, and people have developed a multiple way to determine the heterogeneity further in this population. So to remove that. So here it's the different way that people have used to establish the heterogeneity in the LSK compartment. So initially in the 90s, the Weissman group used the Ty11 marker to isolate stem cells from this LSK compartment. This marker is not truly great. It's not expressed in B6, it's C57 black 6. So it's not something which have been which is still in use. Other people have used dye, and particularly the property of a stem cell to exclude some dye, and this was, for example, the UX exclusion system, which was promoted by the laboratory of Margaret Goodell in Texas. And this was also, she defined a side population which was enriched, which exclude all the UX, export the UX out of the cells. And this was enriched for stem cells, hematopoietic stem cells. This also, for a long time, I didn't see an article using this way of characterizing stem cells. It's certainly due to the fact that it's relatively tricky to put in place, and that the UX has a very broad emission spectrum, and so it's relatively limiting if you want to do co-staining. The other way to isolate stem cells in this LSK compartment is to use reporter mice, which are associated with the expression of genes which are specifically expressed in stem cells. And you have a series of mice which have been developed over the years, reporter mice for OXB5, PDZK1, IP1, and FGD5. None of these reporter is actually perfect to pin down the hematopoietic stem cell compartment, and they are limiting because each time you need to have the strain to do your study. So the most effective way to isolate the stem cells is through a cell surface marker. And so different nomenclature and protocol have been put in place over the years. So what appeared first in the years 2000 was a negative marker, such as FLT3 and CD34, and the double negative for population, or the double negative for these two markers in the LSK compartment is quite enriched in immature stem cells. The difficulty of this staining is that it's a negative marker, so there is a little bit of difficulty to assess lines sometime and segregate positive to negative in this system. The big change was introduced in 2007, I think, by Sean Morrison in his laboratory, which introduced the notion of SLAM code. So SLAM stands for Specific Lymphoid Activated Marker. And the Morrison lab showed that stem cells in the LSK can be isolated based on two of these SLAM markers. They are positive for CD150 and negative for CD48. And this is becoming the dominant way to isolate stem cells in the LSK compartment. In addition of this marker, other markers have been used to further enrich even mature. Hello? Yeah. So other markers have been used to further enrich on the most immature stem cell compartment. So this marker include EPCR, which is also CD201, CD49B, CD105, CD244, and CD229. These are basically an add-on on the stem cell compartment defined by the SLAM marker. So this is all the way that we have to isolate stem cells in the LSC compartment. For the multipotent progenitor in the LSK compartment, you have actually two nomenclatures which are dominant. One nomenclature has been developed by the laboratory of Andreas Trump in Germany, and is based on the expression of FLT3, CD34, and the two SLAM marker 48 and CD48 and CD150. This nomenclature define a multipotent progenitor 1, 5, 2, 3, and 4, which are defined, which are based on this marker. The second nomenclature, which is still in use in many labs, is the nomenclature developed by the Morrison lab in Texas. And this is a pure nomenclature based on the SLAM marker. And so this nomenclature define MPP1, 2, 3, 4, and HPC1 and 2. It's important to note, and it's a little bit confusing in the literature, that there is no truly a correspondence between these different appellations. So you need to be careful each time to verify how these different populations are called. Okay, so I apologize for that. So this was the different way to assess the heterogeneity in the LSK compartment. Similarly, a great effort have been done to assess the heterogeneity of the lineage committed HSPCs. So the classical nomenclature have been developed by the Weissman lab. Again, it's 25 years old, but still, I think, valuable, which is based on the expression of CD34, Fc gamma receptor, IL-7 receptor, and FLT3. And this define common myeloid and common lymphoid progenitor, and also a bipotential progenitor for the granulomacrophage and the megakaryocytic erythroid pathway. This is obviously a very broad and very inaccurate way to separate this population. And so there have been different new nomenclature which are coming to try to deconvolute this different compartment. The most notable are the one developed by the Brider lab, which deconvolute the CMP and MEP mainly in the erythroid and megakaryocytic pathway. And this is based on the expression of CD41, CD150, Fc gamma receptor, and CD105, which is endoglin. More recently, the Goodrich lab have deconvolute the CMP and GMP based also, so this time on the path toward the granulomacrophage pathway. And this is based on Ly6C, CD115, CX3, CR1. And also this is a relatively new and interesting way to deconvolute this pathway. And it's maybe more anecdotic, but if you are interested in lymphoid development, the Weissman lab has also deconvoluted a little bit the CLP based on the expression of Ly6C. This is just a snapshot of what exists, but there is many more way to deconvolute this different population and assess the potential of this or isolate or analyze this lineage-committed HSPC compartment. So here, as we analyze this different population, we decided to design a single complex staining, which include a couple of this nomenclature. So the goal was to provide a comprehensive picture of the early hematopoiesis, ready-to-go staining that we can use all the time to assess the effect of our experiment on the normal hematopoiesis. So we combine several nomenclature developed by a different lab. And we also use this staining to compare a classical flow cytometer with our new Aurora spectral flow cytometer. So this is the staining that I was talking about. So it's a 14-color staining, which include a lineage to exclude all the lineage-positive cells and focus on the HSPC compartment, a life and death discriminant, which is the zombie, and this different marker, which includes c-KIT, SCA1, the SLAM marker CD48, CD150, the marker CD34 and FLIC2 or FLIC3, the IL-7 receptor, Fc gamma receptor, CD41, CD105, EPCR, and CD49B. So with this 14 color, what you have, you have a directly access to the nomenclature of the Trump lab, which describe a basic HSC compartment and several multipotent progenitor compartment. This also allow us to decipher even more this HSC compartment by using the other markers which are available and described in the literature, such as 49B, CD49B, EPCR, CD105, and CD41. So this is for the upper part of the hematopoietic system. In the lower part, this staining allows us to have access to the lineage-committed progenitor defined by the laboratory of the Weissman laboratory, so CLP, CMP, GMP, MEP, and also the nomenclature defined by the Brider lab with a deciphering of this different compartment, at least in a two compartment. So this indicate that with 14 color, we have directly access to 16 hematopoietic population, which encompass all main blood lineages from the myeloid, erythromegakaryocytic, and lymphoid with the CMP. This staining has also built-in redundancy for the HSC compartment. How you can compare the stem cell compartment as it has been defined by multiple laboratory in the literature. So I'm going to give you a very brief sense of how technically this staining happen. So this is a protocol that I learned, and actually it's very much the same protocol that the protocol I learned during my postdoc in the laboratory of Emmanuelle Passeguet when she was at UCSF. So for when we analyze mice and analyze a cohort of mice, we recover the bone marrow cells by flushing femur and tibia from the two legs. We perform a red blood cell lysis using ammonium chloride potassium buffer. We wash the cells to remove any excess of this buffer, count the cells, and for these 4 bone, we expect between 60 to 80 million cells per mouse. We perform our staining on 7 to 10 million cells, and the staining protocol is a 5-step protocol. So for the lineage, we use unconjugated rat antibody. So it's something that can be adapted it's something that I particularly like because I think it's a nice separation. So we use a panel of antibody which are specific, lineage specific. So we use TER119 for the erythroid cells, MAC1, GR1 for the granulomacrophage cells, B220 and CD5 for the lymphoid cells, B2 and B1. And we use CD3 and CD4 and CD8 for the T cell lineage, and I guess also for some dendritic cells. So we do this first staining with unconjugated antibody, and we reveal this antibody with using a goat anti-rat secondary antibody, which is conjugated most of the time with PSI5. The third step is the main staining, where we use all the conjugated antibody that can be, which are part of our panel. We do a fourth step in case where biotin is used in this main staining to stain to reveal and to couple fluorescence to biotin. And after the fifth step is life-dead discrimination. And we use zombie NAR or propylium iodide to exclude all debris and dead cells. All this, at each step, we watch with staining media, which is a basic buffer solution, which is complemented with fetal serum, bovine serum. I would like to give some tips on this staining. So first, I will advise to apply a gentle condition when you do the red blood cell lysis to preserve all erythroid progenitor. So this buffer will lyse the blood cells, the mature blood cells, but he has also an impact on the progenitor MEP, CFUE, and pre-CFUE. So if you let too long this buffer, or if you are too harsh in the way you resuspend your cells, you will affect the number of erythroid progenitor that you recover. I would like to say also that we use 7 to 10 million cells because our protocol is multi-step. So each time you lose cells, and this we found is the number of cells that we need initially to at the end be able to recover and acquire 2 to 3 million events at the cytometer. As I say, we use this complicated system with unconjugated rat lineage and goat anti-rat. I know that there is possibility to use a commercial lineage cocktail, which are already stained. The difficulty, I think, is that for us is also that none of this commercial lineage cocktail actually is specific with PSI5, which I think I prefer to use as a dump channel. For the life and death discrimination, we use we try different possibility. So we now focus on the zombie NIR, which has a far red fluorescence, which is compatible with all over red marker that on the red laser with all over fluorochrome, but only on the aura. I will not advise to use the zombie in a classical flow cytometry in the NIR. And for conventional flow, we use propylium iodide, which is fluorescent as a PSI5. So we put everything, the lineage cells and the dead cells in the same channel, which is a dump channel. This slide is just to, so this was in case when you do analysis and just have you want to have a couple of million events to see your population. If you want to do sort and to have a quantitative preparation, so we use an adapted protocol. In this case, we extract the cells by crushing the bone. We crush femur, tibia, hips, and arm. And so you kind of dramatically increase the number of hematopoietic cells that you recover in this case. So by flushing, we have about 60 or 80 million cells. Here you can get 120 million. So you dramatically increase your yields for ultimately after sort. We do red blood cell lysis as previously. We had a two-step for the sort. We do a purification by density gradient to remove all the debris, and bone residue that have been generated by crushing the bone. And so this is just by a FICOL. And we enrich the cells to limit the time of sorting with based on the kit marker. We use magnetic separation, is a classical Automax Miltenyi, with usually for analyzing stem cells, we use positive selection with the two columns. And in this case, you get about a couple of million of cells per mouse. So we tend to pile up 5 mice to get enough stem cells when we sort. If not, the protocol remains the same in this condition of sorting. In terms for the analyzer, we used two type of analyzer. So a conventional cytometer, it's a Benton Dickinson Fortesa in classical configuration, which is available in our facility. And but recently, since 2020, we shifted to the spectral flat cytometer, Cytek Aurora, which is a 5 laser channel with multiple 5 laser cytometer with multiple channel, which allow to have a pretty decent spectrum for each fluorochrome. So this is our panel. So it's 14 color. The color we limit on 4 laser. We didn't use the UV laser for this staining because at the time where we set up this staining, our cytometer was only on only 4 laser. So this is a different step, a lineage cocktail, which is based on purified rat antibody. This cocktail is revealed by a secondary antibody, which is goat anti-rat. We have a main staining, the addition of streptavidin as a fourth staining to reveal the biotin, and after a life and death marker which terminate the stain. Again, we use zombie NIR for the spectral flow cytometry and propydium iodide for classical flow. So here I put also the dilution that we use in our protocol. I will strongly, it's for indication, it's indicative only. I will strongly advise if you wanted to use it to titrate your antibody and to be sure that you use a good dilution for this staining. So we use a 4 laser. I would like to also to indicate that it's important that you look at the clone for each antibody. So this, for example, for CD150, we use this particular clone. I know that in the market there is another clone, but it's not giving exactly the same type of staining. So I will be cautious on that and at least be consistent with the type of antibody that you use, especially for this marker. I also highlight the CD34 marker, which is an old marker which has been used for 20 years now, but in our hand is not an easy marker. So when we use this antibody, we use at very high concentration, when in 25, and it works well, especially for FITC. So it's FITC is our fluorochrome, our best fluorochrome for this marker. I know that, and we have used when needed other color. We have used BV 421 and eFluor 660. So we make it work, but it's with high concentration like here, and even we need to extend the time of staining. So it's relatively difficult to handle in other color. But FITC is the best if you use high concentration. So this is the staining that we obtain. I will go through that. So this is on the Aura machine by spectral flow. So we use a combination of gating on the forward and side scatter to eliminate debris and to focus on single cells. After this different gating, we arrive in the lineage live cells. So we focus and gate on the lineage negative cells, which are alive, so zombie negative. And after we arrive in this LSK kit SK1 gate, which define the LSK and the LK. So with our 14 color staining, we are able to differentiate here in the kit low, SK1 low gating here. We are able to detect the CLP as defined by the Weissman lab, and also the MEP, CMP, and GMP. We were also able to visualize the population defined by the Brider lab. These include the megakaryocyte progenitor, the pre-GM, another type of GMP, which is a subfraction of the GMP of Weissman. This is the erythroid megakaryocyte progenitor, CFUE, pre-CFUE, and CFUE. So this is for the committed progenitor, in the LSK, for the multipotent progenitor. We have the panel defined by Andreas Trump. So this is the MPP4, the stem cells, which are CD48 negative, CD150 positive, the MPP5, the MPP2, and the MPP3. This staining allows us to further segregate in this CD150 positive HSC based on to segregate long-term and short-term HSC based on the expression of CD34 EPCR or CD34 and 49B. So you have a multiple, as you see, multiple gating, but at least we have a large panel of population that can be analyzed. And if you have a mutation, you can pin down quite a bit where the main effect of your mutation happened. So this was analyzed in the spectral flow cytometer Aurora. And we did the same staining by a conventional polychromatin flow cytometry. So this is a big word to say that we use the Fortesa. So we have the same gating, but I will not go over this population. But overall, it works in this conventional flow. I would say that it's a little bit less prettier in this case than the spectral flow, but yeah, all the population and the percentage are relatively equivalent. So to give you an example, a couple of examples how we can use this staining. So this is, we have done analysis in aging. So we analyze 2, 4, 8 weeks old mice, so a juvenile and adult mice, and a middle-aged mice, which is a 12 months old mouse. Again, we gate on the LSK and we define the stem cells based on CD48 and CD150. And as described by Over, what you see is that you have an increase of the phenotypic HSC compartment in the middle-aged mice compared to the juvenile and the young adult, which is consistent, which have been described in the literature. Also, this aging has been associated with myeloidization of the HSC compartment. And one marker which has been associated with this myeloidization is the CD41. And when you look again in these stem cells downstream in the system of the expression of CD41, you see that this marker is not expressed in juvenile mice. You see some level of staining in adult mice, and you have a dramatic increase of this CD41 marker in HSC of middle-aged mice. Just to say that the system works as planned, and we can reproduce what have been described by Over with different staining. We have also used this panel to look at transcription factor reporter mice. And this was done on the spectral flow cytometer Aurora, which have the interesting ability to differentiate between GFP and Phe. So we use exactly the same staining that previously, and we just add reporter mice which have a knock-in GFP in the GFI1 locus. And this was a gift from Ligram, the laboratory of Ligram at Children's. So GFI1 has been known to be important in the granulocyte differentiation. And consistent with this idea, we found that the GFI1 is expressed in the GMP, the CMP, which are mixed between two lineage or different lineage, is heterogeneous for the expression of GFI1, while the MEP are completely shutting down this locus. We found that, so this is based on the Weissman staining. In the Brider staining, we found exactly the same thing. We look also at the expression of GFI1 in the multipotent compartment, in the LSK compartment. And consistent, which have been described, we found that GFI1 is highly expressed in the most quiescent stem cells, the most immature stem cells. And this is consistent with a report that indicated that GFI1 is important in controlling quiescence in the hematopoietic stem cells. So this was for reporter, and here we use as a control the white-type mice, just to be sure. This was to be sure that there was no overlap between GFI1, GFP, and Phe. More recently, we have also combined an adaptation of the panel that I show you with a cell cycle reporter mice. So it's a mice which has a RFP, a red fluorescent protein, knocking in the Ki67 locus. Ki67 is a very well-known reporter from a cell cycle, which is downregulated at the G1 phase and upregulated in G2M phase of a cycle. So when you look at this reporter during the hematopoiesis, you find what could be expected, but the stem cells or long-term or short-term stem cells are relatively Ki67 negative, but the committed progenitor CMP, GMP are all, which are highly cycling, are all MKS67 positive, and the multipotent progenitor, which are intermediary compartment within the hematopoietic system, are basically all positive or mixed as expected. So this is also a good way, I think, to analyze cell cycle in this population, with narrowing down quite a bit on these different subsets. The last things that we have done is to look at the division history reporter mice. So I'm not going to develop too much, but it's VH2B GFP mice. So it's a mice which is able to express a protein fusion between the histone 2B and GFP. This is an inducible system. So during inducible by the addition, by the introduction of doxycycline. So when the doxycycline is present, this protein fusion is expressed and will be incorporated in the chromatin. You stop the presence, you remove the doxycycline, and then all the cells which are GFP now embedded in the chromatid are going to start to divide, and the GFP is going to be diluted based on the rate of division of this population. So this system has been used with a long period of chase, up to 20 weeks. And in this case, you see that all the intermediary and short-term progenitor have divided and lost their fluorescence, and that the more quiescent or slow cycling compartment have retained the labeling. And this is a good way to segregate the population which have the most quiescence at steady state. And we have done that at 20 weeks, but we also explore the use of the system at different time points, 6 weeks, 8 weeks, and 10 weeks. And you see this result. I will not develop that, but it's, I think, an important tool to explore the dynamic of the hematopoietic, different hematopoietic compartment. So I'm going to finish now with a couple of slides about what is next for this staining. So as indicated, I have indicated all these 14 color were on 4 lasers. So we have a UV laser which is completely open, which can be used, for example, in to add some marker like CD45.1.2 to use this marker upon the transplantation. In our hand, this marker on the UV laser are relatively weak, so we use relatively high concentration of antibody. Be aware of that. If you want to use it, the separation of the population is not that great on the UV laser, but it's usable. The other possibility of evolution will be to introduce a new nomenclature in this staining, particularly the nomenclature of the Goodrich lab, which desegregate or deconvolute quite a bit the CMP and GMP compartment. So this, with the new marker which are coming on the market, should be relatively easy because we just have to add actually 3 markers in our staining. It will be the Ly6C, CD115, and CX3 CR1 to get access to this progenitor in one staining. The other possibility of evolution will be on the erythroid megakaryocytic progenitor, and this is a staining which has been developed by the Sokolowski lab, which could be an alternative or a complement to what has been done by the Brider lab. Again, this will be relatively easy to add to our, should be relatively easy to add to our staining, which it's a couple of markers, CD155, CD49F, and CD71. So 3 markers we can have access to this population. Okay, so I have been long actually, more than I expected, but I'm going to finish here to give, and I give you these 2 papers. It's a little bit of self-promotion. If you are interested in this 14 color panel, I will advise that I will refer you to this paper that we published in 2020, which is in cytometry part A. If you are interested in the use of H2B or Ki67 reporter mice in the HSPC condition, I will refer you to this newly published paper in GXMed. I'm going to stop here. If you have any question, I'm available. If you are interested in one more detail, you can also email me. You will find my address in one of these 2 papers. Thank you, and I'm available to any question.
Did you say these papers are available, downloadable, or?
Yes, they are all online, yes. The 2 papers are online.
Oh, okay. So the full text is available? Or?
Yes, it's available, yes.
Thank you.
If you have question, a technical question, all these staining are relatively simple to put in place if you know the tiny tricks. So if anybody has a question, I'm truly available to give this protocol or the trick that we use.
Is the presentation today available as a PDF anywhere or even PowerPoint after?
I think the presentation is going to be put online. I can send you the PDF if you are very interested with all the slides.
Oh, okay, great. Thank you.
I will also advise to look at the cytometry. We have all the percentage. The reviewer asked us to put even the absolute number that we detect. It will give you a ballpark of this different population, the size of this population, but it's very variable. I have to say, depending of the age, sex of the mice. I'm pretty sure also the condition of Huntsman-Reese are certainly affecting, on the margin the size of this population. But at least it can give you a sense of how big or how small these populations are.
Thank you.
Okay. Okay, if there is no more question, I think we can stop here. Thank you very much for participating, and again, I'm available if somebody has a question. Thank you.
Thank you.
Thank you.